Hi, and welcome to Meet the Microbiologist, the podcast about the people who discover, innovate, and advance the field of microbiology. I'm your host, Ashley Hagen, and today I'm joined by Dr. Nicole Dubilier, who is the director at the Max Planck Institute for Marine Microbiology in Bremen, Germany. Welcome to the show, Nicole.
Thanks, Ashley.
We're
nice
so
to
happy
be
to
here.
have you. So you head the symbiosis department at Max Planck Institute, and your research centers on understanding the importance of diversity and function of symbioses between bacteria and animals. As one of the leading experts in this field, I'd love to hear how you personally describe or explain the concept of microbial symbioses.
Maybe I'll start with how others describe it
Sure.
and how I
That
see
would be
it.
great.
It was originally described as those that work on symbioses know about 100, and my numbers are always off, but over 100 years ago by Nicole Dubilier, who did a very nice and very broad characterization of saying that it is just the living together of two or more different organisms in close physical intimacy for a longer period of time. And the beauty of that definition is that it doesn't imply if the interactions are beneficial or if they're detrimental or commensal. And now we know through sequencing, through looking at the human microbiome, everything, that can depend so much on the host itself, its immune system, and the microorganisms themselves. You know, you take up one nasty gene like EHEC or E. coli, or that can give you a nasty bug. So that is the definition that I like to adhere to.
Wonderful. So why are these relationships so important? And why is this something that you're passionate about?
I'm passionate about it because when I was looking for a topic for my Ph.D. and I wanted to do marine biology, and I definitely was not interested in marine microbiology, period. My PhD advisor said, I'm working on these little worms. They don't have a mouth or a gut, and they live off of symbiotic bacteria. And I still wasn't so sure, but then he described that their main distribution areas are in tropical and subtropical areas, and that I could go collect them. And so that was definitely a motivator.
Very cool.
What motivates me now, though, is that it's been such an incredible time in microbiology because now we see how widespread beneficial interactions are, and we have the tools through sequencing and imaging now to understand them. So that just, every conference I go to, there's a new method to look at an uncultivable microorganism, which is what most of these organisms are.
Yeah, that's so exciting. And I am really interested in digging a little bit deeper into the research missions and some of your expeditions to understand a little more about what you do on a day-to-day basis, because I can understand the draw as an early young professional to being able to go and collect some of these organisms. But to begin with, before we jump into that, I'd love to know a little bit about when you started this research path. What were some of the main questions driving the field at that time? Because you've mentioned a lot of advances in technology. So I'm curious, at that point, what were some of the questions you were trying to resolve?
I started working on symbioses in 1992. As a postdoc, I joined the lab of Colleen Cavanaugh at Harvard University, who was one of the first researchers to work on the symbioses between hydrothermal vent tube worms and their symbiotic bacteria. And when we started, our first and foremost question was, who are the symbionts?
Yeah.
Using 16S and spending a year and a half to sequence a single gene to get that information, spending another year and a half trying to label it with fluorescent probes and show that it was really the symbiont, and then spending what felt like years trying to get the paper out. So that was where we started, and that's also why I emphasize so much how exciting it is that we now have the tools to look at hundreds and hundreds and hundreds of individuals with thousands of genes.
Yes, absolutely. So your research has, I would say, caused a paradigm shift in their understanding of the diversity of species that are the symbionts. So when you're starting with that question of who are the symbionts, How do you begin to even form these hypotheses and decide where to look?
Oh, we're not hypothesis driven. I actually really like to emphasize that we're discovery driven, that what we do is discovery based research. And sequencing is nothing else but discovery based research. And then we look at what we get in terms of results. We get predictions, and then we dive in deep to see, are those predictions correct? But I'm a big defender of discovery-based research because there's such an emphasis on you have to have a hypothesis. But I no longer, I don't agree with that.
Yeah, I love that. So where do you start to look for these discoveries?
Well, mainly you take whatever organism you're interested in and you homogenize it.
Yeah.
And then you sequence the hell out of it. That's what, at least what we always say we do. But then we're actually, we try for the very same individual to save one piece of the host to use for looking for actual visualization through microscopy. And the reason that it's so important for us to have these paired studies is because we do see a lot of variation from individual to individual.
Is there a particular organism, host organism, that you are most interested in or you have found to be the most dynamic in your research?
It's probably whatever I'm just working on, together with my team, what we're working on right now and what we think is the most exciting. Our main hosts are these tiny gutless marine worms that I began my work on in 1992. It was a field, the hydrothermal vent field research was a field where there were a lot of people moving in. And my great advantage at the time were these worms were so small and you needed so much DNA at the time to work on them that nobody else was working on them. So I had an organism where I didn't have any competitors. The disadvantage was nobody cared. So our citations for work, when we still to this day on the gutless worms are not so great. Then when my lab was established and there were other possibilities, I started working on deep sea mussels that occur at hydrothermal vents and seeps, and there are quite a few labs working on those. But and we get more citations just because the field is bigger. But I would say after that, anything that has chemosynthetic bacteria, I'm excited about.
Very cool. So let's talk about the specifics of how you collect these samples and some of these research expeditions.
Yeah, I wish I could show videos.
I do too. I thought the same thing.
So what we do when we collect our shallow water hosts, and I really encourage anybody who goes diving or even snorkeling or just wading out in knee-deep water from in the Mediterranean and the Caribbean. In fact, even off of the coast of Florida, we recently collected them. And what we do is we take some sediment, we put it in a bucket, and then there's a very fancy method called decantation, which means you just stir the sediment around, and the worms, the hosts, are lighter than the sediment, and you pour it through a sieve. And then in your sieve, you've got lots of animals. They're called myofauna because they're small and they're in the sediment. And those that are white, those are our hosts. And they're white because they have sulfur-oxidizing bacteria. They have sulfur vesicles and PHA, and that makes them look white. So that, of course, is great fun because, for example, we're planning a trip right now to the Great Barrier Reef, and we're doing it right. This time we're going to go there in the Australian summer. And it's really rainy and nasty in northern Germany. Then the other obvious collection is when we go out with research vessels and collect our animals in the deep sea with remote operated vehicles or submersibles. And that's a totally different sampling experience. It's crazy because you spend 6 weeks at sea, but you can't get into the water. But it's also another type of expedition that I love.
Very, very interesting. So if you have some of those videos that you can send to me after the show, we'll link them in the show notes, so our listeners can view those, which will be wonderful. So once you've collected these samples, you take them back to the lab, and obviously you're sequencing them. What are you hoping to discover? What are you searching for? And what are some of the key findings that you're working on right now?
Yeah, in the beginning, we were agnostic in that we just said whatever we find
so
is
cool.
fine with us. What we care about is that we find it in more than one individual. If you're looking for a symbiont that hasn't been described yet,
Yeah.
then one of the giveaways is that even if it's in low abundance, if it's regularly present in every single individual or almost every single individual you look at, there's a good chance that it really is meaningful. That said, we've just started finding some very, very cool bugs that we think might actually be pathogens related to Wolbachia that cause sexual, that can cause changes in sexes and insects. They're super low abundant. We can barely find them, but they're so interesting. They have 100 kb genomes. They're teeny tiny, and so we've started looking at them as well. But so generally, we're agnostic. We know they should be chemosynthetic bacteria. That's what we're looking for. We find them. But beyond that, whatever else is there is also really fascinating for us.
Very cool. Let's take a little bit of time to describe the chemosynthetic bacteria that you're looking for, some of those processes and how they benefit the host.
Yeah, if this were my class, I'd say, who wants to raise their hand and describe chemosynthesis? And it's amazing because anyway. Okay, so photosynthesis is you use the energy from the sun to fix CO2. In chemosynthesis, you're using chemical compounds like methane, like hydrogen sulfide. Bacteria can use, and only microbes can use that energy with an electron acceptor, often oxygen, to gain energy and then to fix CO2. So there are only two forms of primary production on this planet. One is photosynthesis and the other is chemosynthesis. So that's it in a nutshell.
Yeah, but this is happening in environments where sunlight is accessible, right? So this is still in coastal environments as well as these deep, more, I guess, challenging environments for survival.
Right. So microbiologists knew about chemosynthesis for hundreds of years. Vinogradski, the Vinogradski column, I'm sure everyone's done it. They're great fun. What they didn't know is that they would provide enough energy and biomass to host large animal communities. And that was what was so remarkable when the hydrothermal vents were discovered, was that everybody assumed there just wasn't enough primary production in the deep sea because so little trickles down from the surface from photosynthesis to feed large animal communities. And so when they discovered them, it's relayed that Bob Ballard can tell the story much better than I can, but that they called up to the Atlantis mothership. Is there a biologist up here? Because, I thought there were no animals in the deep sea, and they're just all over the place here.
Yeah.
What was so remarkable was that we had to go to the bottom of the sea to realize that they're actually widespread, these symbioses in shallow water environments. So it was kind of backwards looking. Zoologists had always known that there were animals that had reduced digestive systems in shallow water systems, in clams and solomia and tube worms, very, very thin ones. And they assumed that they lived off of organic material, dissolved organic material. But that isn't enough really to feed animals. And so really it was literally I know because my Ph.D. advisor who heard about the vents and went, oh, maybe I should go back and look at my worms and see if there are any symbiotic bacteria in there. So he had seen weird structures before, but he didn't connect the dots. And that's just so typical in research anyway, that it takes you a while to connect the dots, and somebody makes a discovery, and then you go back and say, oh, that could be the same for my system.
It's so neat. I love the exploration here and the discovery with the research that you're doing. So you mentioned that you start out kind of agnostic. But then once you have a discovery of interest and you're looking kind of for it to be either the symbiont to be common to given individuals within a host species, what do you do from there? Are you narrowing, based on that discovery, narrowing your interest?
Oh, well, that's just the beginning. Once we know who's there, we want to know what are they doing and how are they interacting.
Right.
And we've been incredibly fortunate in that that we've discovered some cool things that they're doing that we really hadn't expected. And of course, using then transcriptomics, proteomics, metabolomics, and all sorts of imaging methods, we can really dig in deep and really say, and discover pathways that we knew nothing about before. As an example, when Manuel Kleiner was a Ph.D. student in my lab, he's now a professor at North Carolina State University, he discovered a protein that's involved in using carbon monoxide as an energy source. And we were like, carbon monoxide, animals, they that's not a great energy source, but it turned out to be true, and we were able to show it. And so that's the approach we use, is to first see who's there, what are they doing, and how can we prove it.
Very cool. And what are do you have any other examples you'd like to share of some of the neat findings of what they're doing?
All right, but now you don't need to go to any of my other talks today, because that's all what I wanted to talk about.
Shared that.
No, it's fine. One of the discoveries that I was very deeply involved with and enjoyed greatly was when the paper came out 2012, so it must have been in the late, and now I remember, it was a cruise in 2005 with a German research vessel, and we were going to vents on the mid-Atlantic ridge, and the geology there made it very clear that there would be very high hydrogen concentrations. And hydrogen's a great energy source, that there's bacteria, microbes just will suck it up. And I actually hypothesized that the sulfur oxidizing symbionts could be using hydrogen. That was probably the only hypothesis I made, not quite, but it was one that I made with no evidence whatsoever. And there was somebody measuring hydrogen in the gases in the neighboring lab. And I walked over and I said, can you try to see if they can, if you see hydrogen disappearing, if I homogenize the gills where the symbionts are and put them in there. And it took a long time. It took over 24 hours. I went back the next day several times, and he said, just wait. And then I finally told me, he said, nope, sorry, I didn't see anything. And I was really deeply disappointed. And then as I was leaving the lab, he turned around and he said, you have no idea. They sucked up hydrogen like crazy. And so it took us another 5 years to prove that because it wasn't enough, one experiment. But that was a discovery that I have a lot of ownership for.
Yeah, that's amazing. What about a challenge, something that you found particularly challenging in your line of work or over the course of your career, and how did you overcome it?
One challenge? All right, let me try to... I think one of the biggest challenges, we all know it, is you write a proposal, you put your heart in it, you really work hard, and it gets rejected, or you write a paper and you think it's the best paper ever, and it gets torn apart. I mean, this is our daily lives, right? Not daily, thank God, not daily, but we experience it a lot. And so I think I would say being persistent and just going home and saying, I can't say what I say, and having something nice to drink and just moving on from there. That's a challenge I face continuously, and that I think we all know about, and that I feel actually has been critical for staying it out in my career.
Yeah, I understand that. The resilience I think it takes to do science, and to just choose.
To do anything in life.
To do anything in life. That's a good point. We've talked a little bit about the fact that you have an upcoming expedition to the Great Barrier Reef. How do you choose your new location? You mentioned that obviously the weather is nicer at that time of year, but are there indicators for where you would like to go searching for these symbionts?
Yeah. So yes, it is a mixture of where would we like to go because we've never been there and it's supposed to be beautiful. But also where are there, if it's for the deep sea, where it's never our group alone. We join geologists and chemists, and so you talk to them. You say, "Where are you going? Are there the symbiotic hosts we're interested in? Are they there? Can we do something together?" For the shallow water environments, beyond just where do they occur, we're currently very, very interested in our hosts in the Mediterranean. Mediterranean is kind of cool. I call it the neglected isthmus, the canal, the Isthmus of Panama. We all know that that closed a couple of million years ago, but the Mediterranean was dry 6 million years ago and then refilled. And that means that you're looking at a very recent ocean basin, and you can start to put some numbers on, what are the species we find there and how did they evolve? And that means that we're now consistently trying to sample in the Mediterranean, pretty much along all the coasts. At least the northern coast, the southerns are challenging for many reasons.
So diversity of species would be a draw to an environment.
Well, the diversity, but the evolution behind the diversity is actually the question we're trying to answer.
Understood. So what about in terms of how the symbioses between coastal environments and deep sea environments differ? Are there some ways that you could qualify that? Are you seeing species that are repeated in both environments, or are you seeing that these are exclusive to those environments?
So as a rule, it's very different hosts in the deep sea. Although, for example, let's take the mussels, the Bathymedalan mussels that we and others work on. They can often be found in more shallow water environments. And Dan Distel, who was actually one of my postdoc advisors and taught me so much, developed a hypothesis called stepping stones for mussels, where they actually use stepping stones of environments and shallow water environments to then slowly migrate over evolutionary time down to the deep sea. That has proved true. But let's take our shallow water hosts. We have never found them at vents or seeps, although we look, and we don't know why.
But the microbes, are they completely different as well in those environments with the hosts?
Yes, but we don't, for example, see a clade of symbionts that are only present in deep sea hosts.
That's what I was wondering.
Clade that is only present in shallow water hosts. What we see are lineages of bacteria that were able to form these symbioses, but it's in convergent evolution in parallel times.
Interesting. Very interesting. I would also love to hear from you just a little bit about, do you have a favorite expedition? Anything that the coolest thing that you've seen? I think there's so much uncharted territory, and it sounds like you have seen so many places and so many unique organisms that most of us may not have seen in our lifetime. So do you have a favorite? Do you have something that stands out as really interesting?
Besides the parties. So you should know German vessels are not dry, so we're actually allowed to enjoy ourselves every once in a while when we've gotten all our work done. But I would actually, the expedition I just described with hydrogen and the discovery and being hands-on with coming up with this idea and then actually proving that it was the case, that's up there. It would rank among one of my favorites.
Very nice. You talked also a lot about how genomic sequencing has been instrumental in expanding our capabilities in this field. Do you have any thoughts or predictions about what you think might be the next big leap in technology for the field of microbial symbiosis?
We're actually right in the middle with long-read sequencing, and being able to sequence nanogram amounts of DNA, that is just, and RNA, that is revolutionizing our ability to look at non-cultivable microorganisms. The first time that we wanted to sequence our gutless allelochetes, this was with the Joint Genome Institute, and we had gotten a grant to do it. Tanja Voike had just started there as a young postdoc, and we needed 200 worms to do Sanger sequencing. And it turned out it was actually 2 host species. It was a miracle that we could get anything out of it. We can take a single worm now, we can extract DNA from it, we can do PacBio, hi-fi sequencing, MinION sequencing, nanopore sequencing, and get these fantastic long reads that are super critical, particularly because as microbes become symbionts, they start getting a ton of mobile genetic elements, transposases, insertion sequences, and assembling them is a nightmare. So anyway, this has just been really a fantastic gift.
I want to talk a little bit about any downstream applications of these relationships. So when you're identifying some of the cool things that the microbes are doing, are there applications that you're considering for how we can use those to either for environmental sustainability or for other applications that may be of benefit to the environment?
I was just going to tell a story about a paper we actually just submitted this morning, and then I realized, I don't think you're allowed to. But, well, anyway, I'll look for another example.
Sure.
I do know we didn't pursue this, so we're not on the side of trying to develop patents or get anything out of our research. I do know that one of the discoveries with microbes that can use hydrogen in the presence of oxygen as one that is interesting for industry, because the genes that are used, these hydrogenases, they're very sensitive to oxygen, and it turns out they have one single amino acid that's different. I think it's cysteine that allows them to tolerate oxygen, and so that has become very interesting, both in the symbiotic microbes we work on, but also on others in trying to develop hydrogenases for industrial use.
You're the recipient of numerous prestigious honors and awards, including the 2024 ASM Award for Environmental Research. And this award recognizes an outstanding scientist with distinguished research achievements that have improved our understandings of microbes in the environment. I'd love to hear what you consider the most rewarding about your work.
Working with my team, I really have to say, and particularly working with they're young and they keep me young, and it's just incredibly rewarding to see them grow, to see them go off and get jobs and become academics or go to industry or to non-governmental associations. So it's like you're nurturing hundreds and hundreds of your own kids, and getting to work with these super smart kids. I'm sorry, they're grown-ups, but with these. That's, I find, one of the most motivating and wonderful experiences of my career.
I love that. What advice would you have for future marine microbiologists or someone considering getting into this field or career path?
For a marine microbiologist, I would say don't do it if you think you're going to spend a lot of time out in the water, diving or anything like that. You're going to spend a lot of time looking at your laptop or at a screen analyzing your data. So that's an important thing to understand. But I do think, even for myself, the times are very limited when I get to go out into the field, but they motivate me endlessly. And for some reason, I just find it endlessly more motivating to work on marine microorganisms than on some industrial bug that comes from a wastewater plant. That's just my personal bias. God forbid somebody here works on wastewater plants. Yeah.
That's wonderful. How long do you think you're out on a research expedition generally? If you had to average, is this week long, weeks long?
Yeah, the ones that go to, for example, the Mid-Atlantic Ridge, it takes us over a week with the vessel to get out there, and then we have 4 weeks to work there and a week back. So that's usually about 5 to 6 weeks.
Okay.
When we go out into the field, it's usually only a couple of weeks, but we're actually right now trying to set up a research station on a Spanish island called Mallorca, where there are something like 300 flights a day that would let us actually fly in the morning, do some sampling, and possibly, if we had to, fly back in the evening. It can be anything from a day to 6 weeks.
I've been to Majorca. It's beautiful and known for the pearls, so that makes sense to me why that would be a good location. Anything else you want to share?
You asked a lot of very broad and interesting questions, and it's really difficult for me to come up with anything else. Thank you.
Well, Nicole, thank you so much for your time today and for speaking with me. It's been such a pleasure and really wonderful to have you here. I've thoroughly enjoyed it. Thank you. For our listeners' knowledge, nominations for the 2025 awards and prize cycle are open through June 24th, 2024. So you can find more information about the program on our website at asm.org/awards. And nominations only take 30 minutes, so it's a great way to nominate someone that you respect in an excellent way to honor your colleagues. So we just wanted to call that out. That's it for today's show. Thank you for listening. If you enjoyed this episode, please share it with someone who might also enjoy it. I'd like to thank Ray Ortega for our podcast production. I'm Ashley Hagen, and I'll be with you next time with a new microbiologist for you to meet.